Okay, thank you for the time. Thank you for coming. I'm Paolo Caccharelli from Argentina, and the present work is called Automatic Identification of Go Terms related to ripening fruit of tomatoes based on machine learning. And this work was done by Spetale Flavio, Prata Guillermo, y Elizabeth Tapia Chu. Like background, global production of tomato is the second Solanacean importance around the world, in importance economics, below potato. In Argentina, it has the largest area under greenhouse, and for the production, around the 60 and 70% of this production is for fresh consumption. So there are a lot of traits, and in general, food quality is one of the most important factors around the marketing and the marketplace. So traits like post-harvest stress life present economical importance in different breeding programs. In our case, our breeding programs have two parents, Caimanta and the wild exception LA0722, like parents of the many breeding population in our institute. Rules and Nils well development by incorporation of this wild accession. So sequencing, of course, is widely used around the world, and in our case, transcriptomics sequencing has provided a powerful insight into the molecular chains in fruit ripening. So challenges in this work, it was about the automatic identification of biology for functionalities of genes during the ripening process, important because it detects a list of GO terms, GO terms, and gene-related this process. Transcriptome of these parents and their interspecific growth, F number 1, provide the opportunity to estimate the gene actions. And this estimation gives information on association between phenotypics and genotypics variability in these breeding populations. So the main objectives in this project and work is to identify GO terms on biological process related to fruit ripening, in which genes with differential expression are involved in this process, and to analyze their gene actions. Like methods in our institutions have two genotypes that we harvest in three ripening stage in each plant, and these genotypes are cultivated Caimanta from Salta, Argentina, about Solanum lycopersicum, and the Wallea section of Solanum pimpinellifolium from Trujillo, Peru, and its interspecific cross. So you can see Caimanta fruits are very size, and the Wallea section, the size is around 1 grams. So we have three ripening stage: mature green, breaker, and the rest is red ripe. So we start this project with an RNA-seq in Innova 6000 with TrueSeq library and standard specific with parents, and we developed two pipelines in this case, from raw reads, all the filters, and at the end, with HR, we obtain the list of these genes with differentially expressed. So the second pipeline is about this output and to obtain the gene action. Pass to AgriGo and obtain a list of consensus Go terms. So the first result is about the area and seek in different ripening stage, mature green, like a reference, physiological reference, breaker, and red ripe in the three genotypes. In this case, in Caimanta, there are a lot of, or around 7,000 genes with DGEs between breaker and mature green in the first case, and the second case, 6,922 genes in between red ripe and mature green. In our wallet section, LA0722, in the first case, we have 7,313 genes between breaker and mature green, and in the second case, between red ripe and mature green, 6,929 genes. Finally, the last case, in the F1, we have 2,715 genes between breaker and mature green, and 6,994 genes between red ripe and mature green. So then we have done an arrangement analysis in AgriGo to obtain the Go terms statistical significations. In this case, in the cultivar Caimanta, we obtained 11 Go terms and repeat this analysis in the other genotypes. So obtain, in this case, to the domain, the biological process domain, 11 Go terms in one genotypes, cultivate Caimanta, statistical signification, with the statistical signification, sorry. In the second case, in the Wallach section, 25 Go terms, and finally 6 Go terms in the interspecific crossing. So we did a comparison into the biological process domain between these three genotypes and obtained these 4 Go terms like consensus related with ripening stage. So to estimate gene action relate the ripening fruit, we select 20 genes were randomly selected from the total of the 2,744 genes extracted for these 4 consensus Go terms. And the results show 17 genes with negative overdominance from the parent with the lowest value, and the others show partial dominance towards lowest values. 60% of these genes were related to the value of dominant parent, cultivar Caimanta, and the remaining is around 40% and close to the Wallach section. This is the table or the list of these 20 genes in which we can see the negative overdominance close to the parent, the wild parent, and we detect a new cluster or group of genes related with fruit ripening from gene annotation ITAC of Sol Genomics webpage, like this 1, 2, and 3. So we detect 3 new variants or molecular bases around the ripening fruit. In this case, it's a group of genes that cluster to AP2ERF, called regulated fruit ripening via regulation of ethylene biosystem and signaling. Ethylene is the main phytohormone around the climateric fruit, especially tomatoes. So this is our discover in this project and in this work. So, like conclusions, automatic identification of biological functionalities generate a list of Go terms and identify their biological rules related to tomato ripening with low demand for computational and human resource. Differential expression between genotypes of Go terms was also detected, which allowing the estimation of the respective gene actions and give that estimation of gene action, generate an efficient use of genotypics and phenotypic variability to develop new molecular markers to obtain new varieties or to assist selection on desirable traits and obtain new varieties in our breeding programs. So this is our team with a lot of people, fortunately, and thank you so much.
Thank you, Paolo, for the talk. Any question? Yes, there. Please come over.
Hi, thank you for your talk. I was wondering, how would your algorithm deal with the hierarchy in the gene ontology annotations? For example, when you find two Go terms that are related in a hierarchy, how did you deal with that?
The annotation in the table, in the last column?
No, in the gene ontology. When you find two Go terms that are related in the gene ontology, would you keep them or would you consider having the parent or the specific child?
Yes, we consider all the Go terms in each parent and the cross interspecific cross, and we filter to obtain these Go terms related to ripening fruit and to its consensus between these three parents to then pass the rest of the project of the work with the consensus and apply this information in all the breeding population. Is in general around 200 plants, so this information is important for us to develop a new primers or detect polymorphisms and design new molecular markers and to assist this plant breeding in general. This is the way.
Okay, great, thank you.
Thank you so much.
Any other question? Yes, please. Put it on. No, there is a button.
Oh, there. Oh, hi.
Thank you.
Thanks for the interesting talk. My question is more related to the applicability of the workflow. I noticed that you found different pathways that were related to ethylene biosynthesis, and this is a very known pathway that most crops use to deal with ripening. So I was wondering if you could apply this, for example, to other crops like corn or blueberry, for example. What would be the limitations, or do you have to consider polyploidy into this scenario for each crop?
Yes, this was a fair approach or a fair exploration, but in general, we try to validate in other crops. In general, in this page or group of Solanaceae, There are a lot like potato or other fruit, climateric fruit or with ripening fruit, and try to guess or explore around these genes that do the same in other crops to stay safe in detect the new variants that really important in this process, for example.
Okay, very good. Thank you.
Thank you so much.